Drug binding refers to an interaction between the drug molecule and the biological target. This biological target may be any receptor or any enzyme or any protein, where the drug is going to attach at a specific site and produce its pharmacological action. Hello everyone, welcome back to my channel. In this video, I am going to focus on a new topic called as what is drug binding, factors influencing drug binding, types of drug binding, then what is the importance of drug binding. There are various factors which are affecting the rate of drug binding or the drug binding. Among that, one is the main factor is affinity. How much drug having a strong bond or interaction with the receptor, or you can say the strength of interaction between the drug molecule and the target, that is the affinity. The second factor is specificity, means ability to bind only to a particular receptor or enzyme. Here the drug molecule is very specific to its target site. It will not bind to anywhere, it only targets a particular site and bound to that particular site, that is called as specificity. So this factor also affects the binding nature of the drug molecule with the receptors. Third one, concentration of both drug and target levels. It also affects the binding of the drug with the target receptor. So here, as the concentration of both the drug and target is more, the binding also increases. If the concentration of both the drug and target, that is the receptor, enzyme or protein, whatever it may be, if it is low, means the rate of binding of the drug molecule with that receptor is also decreases. Lock and key system. Structural compatibility, that is lock and key or induced fit mechanism. Here the drug molecule has a specific particular target, that is the system, or you can say the receptor or protein or anything, that is the biological molecule, where the drug is going to specifically bind to its system, and the binding is like that lock and key system. This type of thing is called as structural compatibility, or it is an induced fit mechanism. There are various types of bindings are there. Here there are three columns. In the first column there is type of binding, there is binding type, and the description in the second column, and example for that. The first one is covalent binding. What happens in covalent binding? It is a irreversible form of binding and forms a strong chemical bonds. It will form a strong chemical bonds. Example for this one is aspirin, binding to COX enzymes. Second one is ionic binding. So here electrostatic attraction between there. So here electrostatic attractions between the oppositely charged groups. Example for this, atropine with muscarinic receptors. Third one is hydrogen bonding. It involves hydrogen atoms bonded to electronegative atoms. It involves hydrogen atoms which are going to bond with the electronegative atoms. Example for this category is antibiotics binding to enzymes. Then van der Waals force. So it is a weak and short range interactions due to temporary dipoles. Example for this, diazepam with GABA receptors. Then hydrophobic. They are non-polar interactions in the lipid environments. Example, lipophilic drugs like steroids. Clinical implications of protein binding. So here, why it is necessary to study the protein binding nature of the drug. See, when any drug is administered, it undergoes distribution process. And it may bind with the plasma proteins or it may bind with the tissue proteins. And the amount of the drug which is remaining, only that much drug is ready to produce the pharmacological action, whereas the bounded drug is not able to produce the effect on the body. So that is why it is necessary to study the protein binding nature of the drug in order to predict the onset of action as well as the pharmacological action of the drug. So here, protein binding affects how drug work in the body. It influences how drugs are distributed, then cleared and monitored. Then volume of distribution. Volume of distribution means what? So here, how much drug spreads into the body, tissue proteins compared to staying in the blood. Means it is nothing but a measure of quantity of the drug which is going to bound with the plasma proteins the amount of the drug which is going to bind with the tissue proteins. It is nothing but VD is the amount of the drug which is going to bound with the plasma proteins and tissue proteins. So in the easy way we can say it is the measure of ratio between the quantity of the drug which is going to bound with the plasma proteins and the quantity of the drug which is going to bound with the tissue proteins. Here that is why VD, that is volume of distribution. How the drug is going to distribute in the body. How much quantity of the drug is going to bind with the plasma protein and how much drug is going to bind with the tissue protein. So both the things are measured in VD. It is the nothing but measure of ratio between how much drug is going to bind with the plasma protein and how much drug is going to bind with the tissue protein. So we can write this formula, that is VD is equal to quantity of the drug bind with plasma proteins and quantity of the drug bind with tissue proteins. So now there are two categories of the drugs. That is, the first one is high tissue binding. The drugs which are showing binding nature with the tissue proteins, means these category of the drugs bind in more quantity with the tissue proteins. The drug leaves the blood and enters the tissues, and low blood levels, high body levels, common with fat loving lipophilic drugs. See, high tissue binding drugs, what is the nature means? So these type of drugs will leave the blood and bind with the tissue proteins, means the quantity of the drug which is more with the tissue proteins, that is large VD. The volume of distribution is large. Means when we apply this formula, the quantity of the drug, that is VD, is more with tissue proteins, tissue proteins and less with plasma proteins and less with the plasma proteins. And what is the nature of the which type of drugs is showing this nature means the drugs which are fat loving means they are lipophilic in nature. Example, tricyclic antidepressant drugs and antipsychotic drugs. So these category of the drugs will show this type of action. Then another example is there, that is high plasma binding drugs. They have smaller VD value, means these type of drugs will distribute more in tissue proteins, as compared to plasma proteins. Then these type of drugs stay more in blood. They will not leave the blood. They stay in the blood in the distribution process. More quantity of the drug is going to stay in the blood only. That is, we can get high concentration in the blood levels. That is low in tissue levels. So this type of nature will be seen in hydrophilic drugs. Example, protein bound antibiotics. So then why VD matters? So it is helps to calculate the right starting dose and predict how fast the drug reaches the steady state.